is what we are highlighting. Now, if you want to see how it looks like overall results from gene expression analysis processing to pathway analysis to network analysis, and then finally the pathways, crosstalk of the pathways. And these are three different kinds of cancers which I have selected for benchmarking this software. And you could clearly see these are the common pathways between these three types of cancers and also highly enriched pathways. Now, if you want to see in terms of workflow, the basic analysis, like how many genes were differentially expressed, how many pathways were enriched, and then what are the p-values of different pathways in different cancers, and that means how significant they are, and overall how we could represent with their following p-values. And here it is minus log 10 of p-values, just to scale down for better clarity and presentation in terms of color, so that it could be easily identified which pathway is more significant in which type of cancer. Now, so this is another way of representation. If you want to go for classification of, let's say you want to present the pie chart or some other kind of plotting, depending on how well you want to present. So basically, this is, for example, for one data set, GSE 8218, another one here it is for another data set called GSE 1431. And then I tried to present some of the best signaling pathways in different data sets and different types of cancers. This is just for presentation. So how good results you can present with complete clarity. And this is really highly powerful with respect to Ingenuity or EPA software. It only depends on the people, how much they are interested in some kind of manual coding and command line processing. Now, the same thing could be represented in another way. And now this is kind of final presentation of the workflow. So let's say if you want to discuss in terms of graphical abstract, so targeted disease, you can select high throughput data, you can select, then go for the processing for analysis and integration of the data, then go for phenotypics prediction. And finally, I have also included some more things, that is like docking and MD simulations, and also mathematical modeling of signaling pathways are coming in this stage. So it is basically representing the complete bioinformatics and system biology scopes and application for any kind of biological or clinical or diagnostic results. Now, this is the final results which you could see how better you can present with the same thing. And these are the detailed description, what it represents. This is the data which was published in the paper as a benchmarking outcome for prostate cancer. You could see some of the pathways which are having very strong crosstalks between the pathways have been highlighted. For example, PI3K AKT signaling pathways, MAP kinase signaling pathways, then focal adhesion, RAP1 signaling, adrenergic signaling, and there are some more signaling pathways. This is for one data set. This is for another data set, that is AMPK signaling. And then, by the way, there are few things which you will find common. That is top pathways in terms of crosstalk. They are common in all kinds of data set. Here, this data set is from basically, you can say TCGA, that is complete prostate cancer clinical samples. And here also you could see PI3K AKT signaling, focal adhesion, AMPK signaling, hippo signaling. They are common between these things. So it means the software is predicting quite accurate or very close to accuracy, highest maximum accuracy in terms of output. And this has been published in the paper. Now, this is another level of, if you want to present the pathway-pathway interaction in terms of the I matrix and color, then this is another way, like figure B. Figure A is kind of integrated crosstalk between all the three combined data sets for prostate cancer. This is like figure 3A. This is all the 11 pathways, top pathways are here, and then their crosstalk between the pathways. Figure B is basically representing the same thing, but in another way of presentation. Figure C is representing the same thing, but here all the genes, all the proteins are present. Bigger node size means they are highly important in this particular case. For example, GSK3 beta, YWHAE, ATP1A3, PRKCA, and many more. The MYC, RELA, all these gene proteins are showing the highest significance, highest impact in case of prostate cancer. By the way, you can also predict the biomarkers, drug targets, which is like included in this paper, and then, or in this software. Then you can also see the pathways, all the pathways, and the genes present, in which pathways these genes are present. So this you can see clearly. Okay. Now, next figure is for the same figure, figure 3C. This one is represented here in another way. For example, 2 colors means they are present in 2 pathways. 3 colors or 4 colors means they are present in 4 pathways. So more than 1 color means they are present in more pathways, and that's why its significance will increase. And this is like, for example, MAPK signaling pathway groups, 1 color, that means these proteins are dedicated to that specific pathway. For example, here, this is AMPK signaling, only blue color proteins are here. Here, yeah, like these yellow color. Then there are some more things. Then let's say there are something for focal adhesion. Then you can see hippo signaling pathways, dark green color. So these proteins are dedicated to hippo, but they are not sharing their presence in other pathways. So this is what it means. This gives you the idea how much dominant these particular proteins are. And then you can also discuss by seeing the functions, what are the functions which are mostly affected as a result of alteration in these pathways. Now, you can go for next level, that is system biology study. So you can select some of the pathway components or complete pathway, go for mathematical modeling and simulation of the pathway, and predict the dynamics or protein expression in terms of like you can say x-axis, y-axis, z-axis at different input signals, and then at different time points. And this is the final outcome for the target protein, or let's say responses. This is for another condition. So this is, for example, when we are saying PP, double phosphorylated ERK in case of prostate cancer, and this is double phosphorylated ERK in case of normal prostate. So when there is no cancer, then this double phospho ERK responds like a quick switch or fully adapted. This is like sustained response. That means once this protein is expressed, it remains expressed for most of the time points. Now, the same thing can be validated by using ISC, prostate cancer images, ISCs. And this is clear also from this. So basically our hypothesis is validated in human prostate cancer samples also. And then next part is just for the next step or application. I also targeted some of the top ranked proteins in this particular network by using these 3 drugs, and then predicted that quercetin is most effective in the top 3 proteins of this particular crosstalk network. So next is you can also visualize. These are very basic things which you could visualize in terms of binding places, binding sites, binding groups. And then overall, you can see different way of presentation of your data, crosstalk data, gene network data, everything you can go for. Now, this is the final application for all three kinds of cancers: ovarian cancer, breast cancer, and prostate cancer. This is the complete crosstalk map in these three types of cancers. So this is overall what you can do it. And you can also contact me if you have any kind of confusion for implementation of this particular software. So you are most welcome to contact me. Thank you very much.